Introduce her cat, please, too.
Her cat, I don't know her cat.
This is Trudy.
Well, we all know Astrid. She's been a member of this community for quite a long time. Astrid first came to my attention when she was a doctoral student because I was really interested in knowing the internal resistance of axons. And as part of her work looking at how cables influence measured synaptic electrical coupling conductances, she had to measure the internal resistance of an axon. And so that was important for models that we were thinking of at the time. So, and as far as I can see, one of the best sets of measurements of that under real conditions. Then she, after doing her PhD at the Technical University in Munich with Peter Fromhertz, she moved on to Eve Marder's lab, where she did a lot of different projects, it turns out, and all the way from looking at variability in the morphology of neurons and in the outputs of circuits to the piece of work that really started a revolution in our thinking, which was the 2004 paper, Prinz, and Marder in Nature Neuroscience, which started us all thinking about how degeneracy permeates biology, and in particular the nervous system. We didn't realize that, I think, at the time, just how important that observation was, or maybe Eve did, but I didn't. But it started a new paradigm in research, both on the theoretical level and on the experimental level, trying to look for this degeneracy in the nervous system. I know it certainly influenced my research. And of course, we at Emory were very, very pleased to be able to recruit Astrid here in 2005 as an assistant professor. And she's been with us ever since, working away in her lab and doing mainly these days theoretical studies, including work on sympathetic ganglia with Sean Hotchman in the Department of Physiology. So she's been a valued member of the biology community and participates in some of the most creative teaching we've done during the pandemic. She single-handedly took on our huge majors course in neuroscience and behavioral biology during the heart of the pandemic and did a pretty amazing job. So we have in Astrid a consummate theorist and a wonderful teacher. And it's a pleasure to introduce her to you. Someone you all know, Astrid Prinz.
Thank you, Ron, for this very kind introduction. I will share my screen to my PowerPoint presentation here. Okay. Yeah, so I'm going to tell you the story that most of you already know very well, but I hope that those of you who are kind of new to the field will get something out of it. So I will be talking about neuronal parameter degeneracy, and I will do so in the context of neuronal network and circuit homeostasis. That is referred to by this quote here from Walter Cannon, saying that somehow the unstable stuff of which we are composed has learned the trick of maintaining stability. And I will try to make the argument that parameter degeneracy is a big factor in this kind of stability that we observe in neural networks. So the outline, I will introduce the STG, which I can do very quickly to this crowd, and then talk about parameter variability, experimental and computational evidence for its existence. And then I will move on to describe parameter spaces and solution spaces, and point out structure in these that we observe in these spaces that comes in the form of correlations between parameters, and then I will try to tie it back together in terms of how that might be related to homeostatic regulation. So this group does not really need an introduction to the stomatogastric ganglion, so I'm just flashing this up as a reminder that we're working in the lobster and crab stomatogastric ganglion, which can conveniently be dissected off of the animal stomach and be placed in a dish where we can then do really long-term recordings intracellularly and extracellularly. And I hope that the students all appreciate just how amazing this is, because I get a lot of envy from people working in other systems who just are amazed by the fact that we can record for several days even from this system. The work that I'm going to talk about is focused on the pyloric circuit in the stomatogastric ganglion, and even though it does contain several other cell types, I will describe work that has focused mainly on the cell types shown at the left here. That is the anterior burster and pyloric dilator pacemaker kernel, the lateral pyloric neuron, and the pyloric neuron, all connected by inhibitory synapses. And this is something that I'm sure you're all aware that I constantly have to point out to mammalian-centric people. These are inhibitory synapses, even though they use glutamate and acetylcholine. And the pattern generated by this pyloric circuit is, of course, the triphasic pyloric rhythm shown at the right here in intracellular recordings, with a typical frequency of around 1 hertz, or a period of about 1 second or 2 seconds, depending on conditions, obviously. Okay, so this is a slide that I was debating whether I should leave in or take out. I usually have this slide when I speak to non-stomatogastric audiences, because they often wonder, why would you study the crustacean nervous system? To this crowd, I don't need to make this point very strongly, but I will just remind everybody that we have the advantage here in the system that we have identified neurons. That is, there is one or a few neurons per type that are identifiable. They have the same synaptic partners in every animal. They show the same stereotyped electrical activity and project to the same muscles in this case, because we're talking, of course, about a pattern generator that controls muscle contractions in the stomach. And so this pattern is very stereotyped, and this will become important when we're talking about degeneracy. We have very clearly defined functional ranges for the output that this system is generating. Functional ranges in terms of period, frequency, duty cycles, burst durations, and so on. And a reason that I decided to leave this slide in here is this workshop that Farzan and Eve and I just attended last week, that was about circuit function and in which there was a strong subgroup of participants that are all, I would say, almost obsessed with the connectome. So I put this up here, and I'm raising this point because I want to make the point that in this circuit, in the pyloric circuit, we are talking about a circuit where we actually have the connectome, right? We know who connects to whom. We know what we know ranges of synapse strengths, and we know what these cells are doing to each other through those synapses, and that is in stark contrast, in my opinion, to what people are trying to achieve in other circuits, including in the mouse, where they are aspiring to establish the connectome, which I've put in quotation marks here, in what I think is kind of almost like a futile pursuit. I want to quote Eve. She was quoted for that statement many times at the workshop. Eve has said that knowing the connectome is necessary but not sufficient for understanding what a circuit is doing. And I want to make the point that in this particular circuit here, we actually do have a chance of understanding what's going on and how the circuit function depends on the connectome. Whereas in a mouse brain, I would debate whether there even is such a thing as the connectome. I think that we can at best maybe in the future establish a connectome of a mouse brain, but that will not be the connectome in the sense that it will not be the same in every other mouse. Okay.
Astrid.
Yes, go ahead. Oh, I should have said, please feel free to interrupt me at any time. I will not keep an eye on the chat because that's too much for me, but please interrupt at any time. Yeah.
So that's what I'm doing. I would even argue that in the crab SDG, we don't have the connectome. We have one or many, in fact, because even synapses are modulated and come in and out and so on. So there's no such thing really as the connectome.
The connectome, exactly. So things vary over time as well, and depending on the modulatory state that the animal is in. So yeah, I think the notion of the connectome is very misleading, but it does persist, as I saw again at the workshop last week. Although people did make some comments about Sebastian Sung's book being kind of overstated, and so people are realizing that things may not be as simple as, you know, figuring out the connectome and we're done. Yeah, I think it is a little bit of a misleading concept. Okay, thank you, Jorge. Okay, so parameter variability. So this really goes back, and it's great that you interrupted me, Jorge, because now I'm quoting some of your own work right now. So here is some evidence from Jorge's work about variability of the very parameters, the very circuit parameters that are underlying this stereotype behavior that I was just talking about, this triphasic pattern generation with a very stereotyped rhythm that occurs in every animal. So this is by now classic, I would say, data from pyloric neurons, from pacemaker neurons and other pyloric neurons. Measuring here, what Jorge did here is he measured the maximal conductance for different types of potassium channels, potassium currents in these pyloric neurons, and he measured that in various animals, every symbol here being from one animal, and And he made this observation that even though the circuit generates a very stereotyped pattern in every animal, it does so on the basis of fairly variable underlying parameters. So you see that these maximal conductances vary 3 to 5-fold in different animals, even though the circuit as a whole produces the same pattern. So this is the simple observation of parameter variability. So in my work in Yves' lab, I wanted to use computational modeling to try and explore how such variable parameters could influence the behavior of individual neurons, and then later also of circuits. And so we turned there to computational modeling in the form of a conductance-based or Hodgkin-Huxley type model with various membrane currents built into the model that were based on measurements in somatogastric neurons. And you have them listed here. We have a fast sodium current that makes the action potential upstroke. We have different types of calcium currents, various potassium currents. Can people see my mouse, by the way?
Yeah.
You can? Okay,
Yeah.
good. So I can use it as a pointer. And we have a hyperpolarization activated current and a leak current. And we're also modeling in the single compartment model neuron the intracellular calcium concentration, which will become relevant later on when we're talking about activity sensing. So we have this model neuron, and for those of us who are not using computational modeling, this is just a quick summary slide to make the point of to illustrate what I'm talking about when I say model neuron. What we're talking about is basically a bunch of differential equations that reside in the computer and that we're solving numerically. And these equations describe the opening and closing of ion channels. I have here ×8 because we have 8 different membrane currents in this model. And we have another set of equations to describe the calcium dynamics, and then a master equation at the top that describes how the membrane potential changes as a function of these currents. So that's what I'm talking about when I say we have a model neuron. And the approach that I took starting in Yves' lab was really very naive, was to basically say, I don't understand how these different membrane currents interact to generate the behavior of the neuron, the electrical activity of the neuron. So let me just go and do a massive scan of a parameter space and just simulate this model with many, many different parameter combinations and just see what happens. What does it do? And so when I say we scan a parameter space here, what I'm talking about is an 8-dimensional space of maximal conductances. I'm showing 3 dimensions here because that's the best I could do on this 2-dimensional slide. We have a space spanned by the maximal conductances of the 8 different membrane currents that are in this neuron. And so in this first exploration of this parameter space, I varied these 8 parameters that are the maximal conductances of these 8 different membrane currents, and I varied each of them from 0 to some physiologically reasonable maximal value in equal increments. And that basically spans a parameter space of, in this case, about 1.7 million different parameter combinations, because I allowed each of these 8 maximal conductances to take 6 different values. Those of us who have been in the field for a while are probably half asleep at this point, because this is really work that's from quite a while ago, but I think I should describe it because it really kind of laid the foundation of later work on parameter degeneracy. So what I did then was I built a database of model versions that were spanning this 8-dimensional space, and that basically examined what is the model neuron doing for each of these parameter combinations. And here are some examples. So these are examples from this database in which everything is identical in all of these model versions, namely the voltage dependence of channel activation, inactivation, time constants. Everything is identical except for those 8 maximal conductances. And on the right here, you see little colored bars that indicate what is the level of the maximal conductance in each of these model versions. And on the left, you see its membrane potential, and you can see that just by mixing parameters, by mixing these conductances in at various ratios, you get a range of different activities going from silence to calcium spiking. So that is action potentials that occur in the absence of any sodium current. And you have regular spiking that does involve sodium. You get bursting of various types. You also get some non-periodic activity. So just mixing these parameters in different ratios can give you very different types of activity. And because we were interested in repetitive bursting activity in these neurons, because we were trying to understand the stomatogastric, the pyloric circuit and its bursting activity, we kind of went into this database and selected neurons that were bursting in the frequency range that we observe in living neurons, with burst durations that match what we observe in living neurons, duty cycles, that is the fraction of the period taken up by the bursting activity, and various other criteria that were chosen so that the models selected from these 1.7 million database models were matching the biological activity. And if you do that, you end up with models, some of which are shown at the top left here. So here we have 9 models that were chosen to replicate biological activity under various criteria. And we're looking at the maximal conductances for 6 of their 8 membrane currents. And you see that you get a range of parameters, a range of maximal conductances. For example, for the sodium here, you see that ranging from 100 millisiemens per square centimeter to 400 is the highest value here. So that's a 4-fold range, and similar for the other conductances, and we can express that as a standard deviation over the mean, or coefficient of variation here. And it turns out that if we compare these coefficients of variation, they are roughly in the same range as the ones that Jorge saw in his experimental determination of maximal conductances. So this is what I'm talking about when I talk about parameter variability. So we have the same electrical activity generated in every animal and in each of these models on the basis of different mixtures of maximal conductances. So the next couple slides are then summarizing the 2004 work that was taking this same approach to the network level. What we talked about before was just a single neuron, and now we're using the same kind of brute force, naive approach on the network, where we're now varying both the identity of the individual neurons. So we have 5 to 6 different LPs, PYs, and AB/PD models that we can plug into these circuit locations. And we have 5 to 6 different strengths of each of the synapses that connect them. And we're basically doing the same approach, namely just covering this entire parameter space with a grid of simulations. And when you do that, you again find that the circuit, depending on the cellular properties and the synapse strengths, can produce various different kinds of activity. And it can produce very similar activity on the basis of different underlying parameters. So this very busy slide that we put in the paper just shows you exactly that. Namely, we have at the top networks that are identical in their cellular properties, but differ in their synaptic strengths, as shown by this legend up here. And at the bottom, we have cells that circuits that conversely are identical in terms of their synaptic connections, but differ in their cellular properties. And in both cases, you can see that the circuit can produce various different activities. So you have, some cases where it fails to generate a triphasic rhythm, with some cells going silent. Actually, that's on the left here. And then you have cases where it shows somewhat irregular activity. You have cases where bursts are skipped. You have cases where you do have a triphasic rhythm, but it's in the wrong order. That's in this column here, where it's not LP, PY, PD, but LP, PD, PY. So wrong order. Oh, I'm sorry. I think this is my smoke alarm going off in my house right now. I think I have to leave. I'm so sorry. Yeah,
That's fine, Astrid. Take care.
Go check it
everything
out.
okay?
Yeah, I will be back as soon as I can. But
Oh, there she is.
You survived.
I survived. Yeah.
Someone needs to edit the recording later.
Yeah.
It was a false alarm, wasn't it?
It was, although a neighbor that I talked to out there, she said she did smell something on the first floor, that some smoke smell. So maybe it wasn't completely for nothing, but the building is still standing.
Good.
And I'm amazed that at least some of you are still here.
Oh, no, we told really good stories that you missed.
Oh, yeah. Too bad. And I was thinking I should have taken my computer with me and just lectured from outside, but too late. Okay, so should I pick it back up, or do you have more stories to talk about? Okay.
You might have to speed up a tiny bit.
Yeah.
Yeah,
I will try. It's mostly old news anyway. Okay. So here we go. I was just talking about this network database where we also observe that varying parameters can lead to different activity, and including, as is shown in the two right examples here, this one and this one, including activity that matches the biological activity in all kinds of criteria, burst durations, period, spikes per burst, and so on. So the bottom line then is that we have, and here are two examples that I will not go into detail on in the interest of time. the bottom line is that at the cellular and at the circuit level, you can generate functional and stereotyped and very similar activity on the basis of different parameters. So there's your non-uniqueness, which I prefer over degeneracy, but I have realized that I have to give in to the community, which has decided to call this degeneracy. So I guess I'll just have to live with that. I don't like the term because it sounds, well, kind of degenerate.
You think it sounds like, yes, it's not.
Yeah. Okay, good. So so now that we have established that biologically and in models, different parameter combinations can produce similar and functional activity, and that gets us to the notion of a solution space. That is, in this high dimensional parameter space, there is a subset of that space that produces a given desirable activity, and we call that the solution space. And a priori, when we started doing this, it wasn't really clear what the shape or the nature of these solution spaces is. So it wasn't clear, are these like small little pockets of parameter combinations here and there that produce functional activity, or is it a one nicely connected and convex blob? It wasn't really clear. And what we have found in the meantime over several years, and what other people have also seen, is that these solution spaces do appear to be contiguous, and but they do not necessarily have simple shapes. So it doesn't appear to be a nice convex blob. Rather, the bottom right here is probably the closest to what I think these solution spaces look like, and that is that they tend to be contiguous, but non-convex. They can have pretty elongated features. So to get at then what these solution spaces look like, we worked with Tim Hickey and John Langton, who are computer scientists, John was and Tim still is, computer scientists at Brandeis, who were interested in helping us visualize these parameter spaces. And we came up with this tool that is called dimensional stacking, which I will very briefly take you through that, because I think it is a useful tool that has now been used by other people as well in order to visualize these structures. So the idea basically is, imagine you had a two-dimensional parameter space. So you have just two parameters that you're varying. And an example of that is up here, where we have the sodium conductance and the calcium-dependent potassium conductance as the two parameters. And if you had just a two-dimensional parameter space, it would be very straightforward how to visualize that, right? You could make a grid like this and then color each pixel by whatever activity you're interested in. So you could make the spiking model versions blue, and you could make the bursting model versions red, and the silent ones green, and so on. So so this would be a two-dimensional parameter space. And the idea of dimensional stacking now is to basically do that for higher dimensional spaces by stacking little two-dimensional grids like that into the next level of organization, where you now have, within this little red square here, you still have the calcium-dependent potassium and the sodium varying, and then you're organizing little squares like that by two other parameters, in this case the delayed rectifier, and I have to move my zoom menu out of the way, and the transient calcium. You're using those additional two parameters to organize those little two-dimensional stacks into into the next level of organization. And if you do that through 2 more levels, then you basically arrive at a dimensional stack that shows the entire 8-dimensional parameter space, like every grid point that I had previously simulated is represented here as a pixel, but they are arranged according to their parameter values. And so you can see the entire 1.7 million database in one 2-dimensional plot. And what that looks like is this. So here's a dimensional stack of the same database that I described earlier. And here we have colored the pixels according to the legend that's shown in this pie chart up here. We have silent versions of the model in gray, tonically spiking models in blue, and then ranging from green through yellow to red, we have bursting neurons, and they are colored according to their number of spikes per burst in this case. And you see a lot of structure there, right? So how do you read this? Let's look, for example, at this bottom left square here that is almost entirely gray. This corresponds to 0 of the highest order parameter here, 0 calcium dependent potassium and 0 sodium. So models that have 0 of those 2 conductances almost invariably are silent. And then another thing that I can point out here is if you look at any one of these squares here, you see this spectrum going from the bottom right in green to the top left in red. So that's a range of numbers of spikes per burst. And apparently what this shows is that the number of spikes per burst increases as you go from bottom right to top left, which means as you go from high transient calcium conductance and low delayed rectifier conductance to low transient calcium and high delayed rectifier. If you make those changes, you walk to the top left here and you increase your number of spikes per burst. So this is how dimensional stacking can be used to basically make sense of these structures in parameter space of these solution spaces. So one thing that emerged both from analyzing these databases at the cellular and circuit level, but also from electrophysiology and from single cell PCR, is that these the parameters that produce a certain type of activity are often linearly correlated. And this is work that you're, I'm sure, are all familiar with, primarily started by Dave Schultz when he was in Yves' lab, and now obviously in his own lab. And so what Dave did there was to look at expression levels of ion channel mRNAs in individual pyloric neurons, and he discovered that if you measure the copy number of ion channel mRNAs in different identified pyloric neurons, you find that they are often linearly correlated. So I know that everybody's familiar with these plots. What we're looking at here is linear relationships between pairs of mRNA copy numbers that are coding for different ion channels, and the existence of these linear correlations and their slope can depend on the cell type. And so from this, and also from what's shown at the left is some of Jorge's data again, electrophysiology. So from these two independent approaches, it emerged that it seems that what defines a cell type in the stomatogastric ganglion is not a particular value for a certain cellular parameter, like copy number of RNA coding for sodium channels, for example. It's not a particular value that defines a cell type, but it is a relationship between different parameters. And in different colors here, you see different cell types, and so it emerged from this data that cell types seem to be can have linear relationships of the same pairs of ion channels, but with different slope. And so it seems that it's kind of this set of correlations or co-regulation rules that characterizes a cell type, as opposed to a given value for each of these conductances. And that is something that I think by now people have accepted this, but initially, as Eve can tell you, and as many of you know, there was a little bit of a reluctance by some to accept this as an observation. Okay.
Astrid, just for the record, that left-hand side graph is from Ron's lab, not from mine.
Oh, okay. Sorry about that, Ron. Okay, so we were then interested in my lab in looking at whether we could find correlations like that also in our model databases. So this is again going back to this database symbolized at the bottom right here of a single stomatogastric model neuron with 8 different membrane conductances and the entire 8-dimensional parameter space covered with a grid of simulations. And what Amber Hudson did when she was a graduate student in my lab is she basically created the model equivalent of a cell type, in the sense that she said, I'm going to pull out of this database all the model versions that produce a certain type of activity. And I'm going to use that as a proxy, as a model equivalent for a cell type. And so what she did here is she first broke down the entire database into models according to their overall activity. So she had spiking models, silent models, and bursting models. And then she broke those down further in many, many different ways, only 2 of which I'm showing here. So here, what I'm showing you is she broke down the spiking models according to their spike frequency. So below 10 Hertz, 10 to 25 Hertz, and so on. And she broke down the bursting models according to their duty cycle, that is the fraction of the period that's taken up by the burst. And when she did that, and she basically carved out subpopulations from this database, and when she looked at those subpopulations, she found that they often also showed linear relationships between their maximal conductances. And what this table is basically showing you here is just the number of correlations that she found in each of these subcategories. So this number 4 here means that among the model versions that produced tonic spiking activity between 10 and 25 Hertz, within that subgroup of the database, she found 4 correlations between pairs of parameters. So to us, that meant that these correlations between cellular properties that we see in the living neurons may have a role in setting up the activity of that model neuron, and maybe a mechanism through which the stomatogastric neuron can achieve functional behavior. And I will skip this because in the interest of time. What I do want to point out is, we're looking at here, as in Dave's data, we're looking here at linear relationships between pairs of parameters. And so because we're dealing with a high dimensional parameter space, the obvious question is, well, what is the shape of that solution space of that group of parameter combinations in the larger, higher dimensional space? And to get at that, what Amber did here is to kind of throw an increasing number of constraints at the activity. So she basically, what I previously showed you, she was just carving out subpopulations that were spiking in a certain frequency range or had a certain duty cycle of bursting. Now she was basically asking, what if I require my burst duration to be in a certain range? That would be down here, this triangle here. And what if I separately require my burst period to be in a certain range? That would be this this triangle here. And what if I now impose both constraints on my models? That is, I'm going to look for models that not only have a burst duration between 0.5 and 0.75 seconds, but also a burst period between 1 and 2 seconds. And that would be this triangle here. And each of these triangles show you what correlations between pairs of conductances did she observe in these selected groups of model versions. And basically, the message of this slide is it's complicated. So it's not like you have one set of correlations between pairs of conductances that ensures that you have a certain burst duration, and then you have another set of correlations that ensures that you have a certain burst period. And when you now require both of these constraints, you get a combination of those underlying correlations. But as you throw additional constraints at the activity of the model neuron, some correlations that exist when you only constrain burst duration go away when you also constrain burst period. Other correlations newly appear. So it's not at all intuitive how this solution space kind of gets shaped as you make more and more requirements on the activity that you want your neuron to achieve. So I am, to be honest, it's a little bit frustrating, but I have for several years now been stuck at this stage where I really do not understand exactly how these solution spaces are shaped as you impose constraints on the activity. And this, of course, may seem like a kind of an academic exercise. We're looking at a high dimensional parameter space of a model neuron, but biology has to do the same thing, right? So the living neurons need to perform a certain type of activity, and that means that they need to certain correlations between their cellular parameters. And how do they do that, right? How do they navigate in this seemingly pretty complex parameter space? This is work by my former graduate student, Quinn Tian, where she basically used the same approach, but she was then examining, I'll go through this very quickly. She was then examining how do correlations between cellular parameters and in this case circuit parameters change when when constraints on the desired activity are made more narrow. So in this case, she is comparing circuit models. So this is these are models of the caloric circuit with the 3 cell types and all the synapses, and she has used evolutionary algorithms to simulate these caloric networks and to evolve in the evolutionary algorithm parameter combinations that produce a functioning caloric rhythm within biological ranges. And she's now comparing a group of model versions that fall within 2 standard deviations of the biologically observed period, comparing that to a subgroup that falls within 1 standard deviation of the mean of cycle period. And what she observes is that as the constraint on the activity that we're looking for in this model, as the constraints are tightened, the correlations also tighten up and become stronger. And this is summarized in this schematic that she made, where it appears that, at least in our hands in these model databases, if you impose very strong constraints on your model, you expect it to perform bursting activity at exactly 1.5 seconds burst duration period, exactly with in a given range of burst durations with some exactly prescribed phase relationships. Then you end up with very few parameter combinations that can actually do the job. But as you loosen those constraints, if you allow a wider range of burst durations of periods, then you start seeing relationships that for relatively narrow constraints still look very linear, but that eventually, when you loosen your constraints, can turn into entire regions in parameter space. So it seems that that basically, in order to achieve a very narrowly defined type of activity, the biological system has to have stronger correlations between parameters. So in the interest of time, I am going to only say a few sentences about homeostatic regulation here. So this again gets back to this question: what does all of this mean for the biological system, right? We see in our models that that tight correlations between parameters can give you a relatively narrowly defined type of activity. But how does the living system navigate in this parameter space? How does it make sure that it maintains proper electrical activity despite ongoing changes in parameters, and how can it regulate itself back into the solution space if it has fallen out of it for some reason due to some perturbation? And there are ideas that Eve has in her lab explored a lot, and that I have also worked on, including I have to skip all of this because of the fire alarm, unfortunately. So the gist of it is basically that we think that that neurons and circuits maintain themselves within the solution space, or can bring themselves into the solution space after perturbation by through a variety of mechanisms, possibly one of which one type of mechanism is activity dependent. So that is, the circuit monitors its own electrical activity through some sensing mechanism and then adjusts its parameters in order to maintain proper activity. And then there is evidence that there are also activity independent mechanisms in which ensure certain parameter correlations independent of any activity sensing. I summed up many years of work by multiple people, including Tim O'Leary when he was in Eve's lab, who came up with this very, I think, very attractive potential model for how this kind of activity-dependent regulation of cellular parameters for activity homeostasis could occur. So yeah, I do not have time to go into any of this. I'm sorry about that. So so to summarize, we have seen that there is parameter variability. That means cells and circuits can produce functional activity on the basis of non-unique parameter combinations. Different parameter combinations can achieve the same or very similar electrical activity. And so we can also call that degeneracy. So the circuit parameters are degenerate in the sense that there is not one unique makeup of a circuit that produces proper activity. If we think about these parameter sets that produce functional activity as solutions to the problem of producing that activity, we can think about solution spaces. And we have seen, and I didn't have time to show you a whole lot of evidence about this, but we have seen that these solution spaces appear to be connected in the larger parameter space, but have pretty complex structure. So it's not necessarily intuitive how they are shaped. We've seen that there are within a solution space, there are often correlations between parameters, and we've seen that in the models. We've seen that in electrophysiology, and we've seen it in mRNA copy numbers. So it appears that correlations are what makes a cell's identity and determines its proper function. And we have, started to scratch the surface in terms of how a biological circuit might actually navigate these complex parameter spaces through activity dependent or also activity independent homeostatic regulation, neither of which I had time to talk much about. Okay, so that's basically it. Work that I briefly talked about and that I didn't talk about because of time was by former graduate students Amber and Quinn Tian, and also by postdoc Cengiz Günay, who is here, I believe, or was before the fire alarm. Still here. He's still here. Okay. The dimensional stacking work was done in collaboration with John Langton when he was a student with Tim Hickey in computer science at Brandeis. And of course, I have to thank Eve, without whom none of this would have happened, because she's really the inspiration and is kind of was and is kind of my guiding star in all of this. And as you all know, can really crystallize insights in an amazing way. So thank you, and thank you for your attention. Any questions? Oh, I see someone had their hand up, but now it turned into a clapping hand.
Yeah, no, that was my bad. Thank you. I have a question. Oh, sorry.
Go ahead. Go ahead.
Thank you for your talk. I am a big fan of your work. I have this question for you. You were talking about solution space, right? As if, so how do you deal with multi-stability, right? It's not the case that for one set of conductances, you have one type of activity. You can have many types of activity in principle, right? Because there could be multi-stability. And also, if you keep adding neurons to a circuit, let's say that you're talking about the model of the full ganglion that has the gastric cells and all of that. Does it make sense to talk about solution space in the same terms?
So first, about the multi-stability, these, you are exactly right. These neurons, in fact, some of them do show multi-stability. We've done some work on that with my former student N. Hong. We published a paper that was exactly looking at, well, it wasn't the main focus of the paper, but it did point out that these neurons can be multi-stable. These models can be multi-stable in the sense that you have, if you inject current to construct an FI curve, a frequency versus current curve, and you do that, you apply a sequence of steps of current injection, you get various frequencies, and if you take that same model version, the exact same parameters, and you step the injection current in an increasing fashion, you get one FI curve, and if you decrease it, you get a different FI curve. So there's some hysteresis there. So for the same, this was a very long-winded way of saying these neurons, these models can in fact be multi-stable in the sense that for the same injection current, they can generate different spike frequencies depending on the previous history. How do you deal with that in parameter space, in solution space? I think that's a very good question. Yeah, you basically you would basically have like overlapping solution spaces, right? Where the shape of the solution space depends on the previous history. I can't really imagine how you would visualize that, but
right, it's super complicated.
Yeah, it's very complicated. So that wasn't a very good answer. It's complicated.
Well, it is what it is. Thank you.
It is what it is. Yeah. So, but again, you have to kind of marvel. I kind of marvel at the fact that with such complex dynamics, even that can even be multi-stable, where even for the same parameters you can get different activity out of the model depending on its history. You have to marvel at how biology deals with that. Like how do you put together a brain that does anything if its components are so tricky, basically.
I completely agree.
Yeah.
I have a question and a comment, or both in one. It seems to me that these solution spaces are really a reflection of our limitations in computational power, maybe because A static picture is certainly not adequate. So these solutions are obviously morphing in real time. They're under modulatory control and whatever, plasticities of all sorts. And we're only looking at conductances. I mean, probably happening at the level of every single parameter that you can think of in every single cell within the huge circuit. So we cannot possibly capture it with the tools that we are currently using, it seems to me. And then that's my comment. The question is you showed that this 2-parameter space where you're restricting the borders, the edges, that seems to align these things better. Doesn't it? I mean, it seems also very limited in the sense that these cells are solving lots of problems at the same time. So you may have a correlation that corresponds to a certain spiking frequency, but at the same time, the cell is bursting and it's doing a million other things for which other correlations seem to be important. So the cell cannot satisfy everything ever at once. I mean, at the same time, it seems to me. So how do you see that? How do you see this problem? It doesn't seem to me realistic to assume that a cell that is as complex as, let's say, an AB cell in the pyloric network can satisfy all the activity constraints or activity features with correlations, because one will break the other. When the correlations that satisfy one will not match or satisfy another.
Yeah, so to that, I have 2 comments. One is, or 2 feeble answers. One is, well, but you showed that they do have correlations, right?
Yeah, but
so apparently these cells can do all of these things and have correlations. So that's one answer. That's just, you know, pointing to your own results, showing that these correlations exist. The other is going back to Amber's figure, which I didn't do a very good job explaining, where, you know, where you constrain the burst period to a certain range, you constrain the burst duration to a certain range, and in each of these cases you do see some correlations. And then when you apply both constraints, some of those correlations go away, some new ones appear. So it's not as simple as as your question was implying, in that you said, you know, to satisfy one requirement, you have these correlations, and then to satisfy another requirement, you have contradictory correlations. It's not a simple superposition of constraints. So I have, that's my frustration that I was trying to express. I have no good explanation for what's going on there, right?
Because we assume that these linear correlations mean the system is sort of linear in some way, but it isn't.
There's nothing linear anywhere. But I think to my mind, the key is that these systems are built to slide between mechanisms smoothly. And by sliding through, you know, so they're not degenerate in having discrete patterns. They actually do very smooth slides between properties so that you extend the full dynamic range of every cell, and it can do switches, and it can change frequency, and it can maintain function. And it's hard to capture the full range of that in a static diagram. You know, in Leandro's paper, where he shows that when you just take a neuron through temperature, you actually switch the composition of the K currents involved in spike repolarization. And it does that smoothly and continuously. And that same observation was made 20-something years ago in Aplysia, where John Kester, I think it was, and his students showed that as you look at different spikes in a train of spikes in aplesia, you actually see different contributions of K currents to spike repolarization. So I think to my mind, that's the key thing about biology, is that you're actually doing sliding, sort of gradual changes in mechanism.
Yeah, so as you see, I'm pulling up the slide that I went over very quickly, or not at all. And I'm doing that, and here's my other cat. I'm doing that because what you just said, Eve, with the sliding between different types of activity, that reminds me very much of this slide here. So what we're looking at here is different subgroups of spiking neurons, model neurons that are selected for certain spike frequency ranges that are given at the left here. And we're looking at a color plot of just these two parameters, the transient calcium and the sodium conductance. And you see that as you decrease the spike frequency that you select for in these models, you get these what seems like linear relationships with high density of models along kind of this wedge-shaped thing. But as you decrease the spike frequency, that correlation seems to be moving through the parameter space. And so I think Jorge was right in that these things are not static, right? And it's very hard to think about how they behave, but it does appear that that the solution space can vary smoothly, just like Eve was describing, can vary smoothly from one behavior to another. In this case, from one spike frequency to another, it can smoothly move through parameter space. Stop share. Okay, are there any other questions? Well, thanks everybody for sticking around even through the fire alarm.
Sorry about that.
Yeah, for which I would apologize, but it's not my fault.
You can still apologize.
I apologize for the fire alarm.
It was the cats.
Anyway,
It was the
bye,
cats.
everybody.
All right.
Bye. Thank you.